consider a patient presenting with type 2 diabetes. Their caloric intake is strictly controlled and their BMI is perfectly normal. Under a traditional caloric model, this pathology makes no sense, but an examination of their occupation reveals the missing variable. They work the night shift. This chart compares type 2 diabetes incidence rates among rotating shift workers against standard daytime workers. Because the data is explicitly normalized for BMI and caloric intake, the elevated risk among shift workers isolates a specific non-dietary trigger. The standard endocrinology model assumes metabolic disease is entirely a problem of macronutrient and caloric excess. It treats the human body like a simple thermodynamic bank account. Calories in, calories out. When we hold calories equal, the timing of food intake and sleep independently dictates the onset of insulin resistance. Human metabolism operates as a rhythmically gated cascade rather than a static homeostatic loop. Systemic insulin sensitivity and postprandial glucose handling are not constant properties. They fluctuate drastically depending on the time of day. This temporal architecture is orchestrated by the suprachiasmatic nucleus, or SCN, located in the hypothalamus. It serves as the master biological pacemaker, taking its primary cues from environmental light. To understand the pathogenesis of insulin resistance, clinicians must look past the gross caloric load on a plate and examine the biological clock ticking inside the cell. Deep inside the nucleus of virtually every cell in the human body, we find the core circadian machinery. It begins with two protein structures, BMAL1 and clock, fusing together. This newly formed complex acts as a primary transcription factor. It docks onto the DNA at a specific sequence known as the E-box motif to initiate the clock cycle. Once bound, it activates the transcription of the period, or PER, and cryptochrome, or CRY, genes, sending a stream of messenger RNA outward. These PER and CRY proteins gradually accumulate in the cytoplasm, fold, and eventually translocate back into the nucleus to physically block the BML1 clock complex, shutting off their own production. This mechanism is known as a transcription-translation feedback loop, and one full cycle takes approximately 24 hours to complete. The scale of this mechanism is massive. This specific molecular loop regulates up to 20% of the entire cellular transcriptome. Circadian rhythms are hard-coded genomic machinery dictating daily cellular behavior. This genetic loop scales up to govern entire organs, specifically targeting the metabolic gating of the pancreas and the liver. In the pancreas, BMAL1 regulates transcription for insulin exocytosis. Beta cells actively release insulin vesicles in a rhythmic pulse synchronized by this local clock. In the liver, peripheral clocks optimize the timing of glucose production and storage, aligning glycogenolysis and gluconeogenesis precisely with the host's active phase. Animal models lacking the core clock gene demonstrate the necessity of this timing. Without it, they exhibit immediate hypoinsulinemia and rapidly develop diabetes. Healthy hormone secretion requires distinct peaks and valleys. A flat, continuous release model results in cellular exhaustion and diminished receptor sensitivity. Pancreatic and hepatic health rely entirely on these local molecular clocks to establish rhythmic tissue sensitivity. Modern environments constantly trigger chronodisruption through shift work, erratic feeding schedules, and artificial light at night. These behavioral cues shatter the synchronization between the master SCN pacemaker in the brain and the peripheral organ clocks. This chart shows the state of forced internal dyssynchrony. We see a sharp synchronized insulin response curve compared against the blunted, delayed response typical of a chronodisrupted state, where the liver and pancreas receive contradictory temporal signals regarding metabolic demands. This internal uncoupling mechanically blunts rhythmic insulin secretion and alters glucose handling independent of the actual calories consumed. This temporal dyssynchrony extends deep into the gut lumen. The gut microbiome is a vast ecosystem that exhibits its own daily compositional shifts. Unlike the master pacemaker in the brain, these microbial populations are entrained primarily by host feeding rhythms rather than light exposure. When social schedules force food consumption during the biological rest phase, it induces a state known as gut jet lag. Gut jet lag immediately alters the microbial structure, shifting the ratio of firmicutes to bacteroidetes and halting the production of beneficial short-chain fatty acids. The physical consequence of this dysbiosis is the rapid degradation of the intestinal epithelial barrier. The tight junctions crack open, allowing toxic particles to slip through into systemic circulation. These leaking particles are lipopolysaccharides or LPS, endotoxins originating directly from the cell walls of gram-negative bacteria. The arrival of LPS in the bloodstream triggers a fierce immune response, activating macrophages throughout the body. This creates a state known as metabolic endotoxemia, a persistent low-grade systemic inflammation. This systemic inflammation directly interferes with insulin receptor signaling in peripheral tissues, blocking the cell's ability to absorb glucose. A gut-derived inflammatory cascade triggered by mistimed eating is a primary clock-dependent driver of insulin resistance. The pathological chain moves in one direction. Environmental temporal misalignment leads to genemic clock disruption, which drives gut dysbiosis and resulting endotoxemia. Metabolic syndrome and type 2 diabetes are disorders of temporal disorganization. One of the most powerful effects of lifestyle interventions is not simply what we eat or how much we exercise, but when we do them. Properly timed meals and physical activity act as potent zeitgebers, synchronizing the body's molecular clocks and improving insulin sensitivity. Clinical insulin resistance is the physical manifestation of a fractured circadian microbiome endocrine axis. Clinicians can address this through diagnostic chronoprofiling. Before prescribing standard metabolic therapies, a physician should assess sleep quality, shift work history, and evening light exposure. This chart maps a consolidated 8-hour time-restricted eating window against the natural peak of beta cell insulin sensitivity and the nadir of melatonin secretion. Time-restricted eating functions as a chronobiological therapy to resynchronize peripheral clocks. Strict alignment of the feeding window stabilizes diurnal hormone secretion, particularly the cortisol rhythm, and rescues pancreatic beta cell sensitivity. By aligning modern clinical interventions with ancient evolutionary biology, physicians treat the temporal root cause of metabolic disease rather than managing downstream symptoms.